Good morning, children. In the previous class, you have studied about the mechanism of sex determination in organisms. Today, let us discuss about mutation. So, what is mutation? The term mutation refers to a sudden inheritable type of change. A sudden change occur in an organism, but that change, if it is passed on from one generation to another generation, that type of change is called mutation. So mutation is a sudden inheritable type of change. So what happens in mutation? In mutation, alteration of DNA sequence occur. Change in DNA sequence occur. Here the term DNA sequence refers to the change in the nitrogenous base sequence of DNA. Clear? There is a change in the DNA sequence, and this change leads to change in genotype. What is a genotype? The genetic makeup or the genetic constitution. So once the genotype is changed, automatically which one also will change? Phenotype also will change. What is a phenotype? The external expression of a trait. You have studied tall is phenotype. There again you have studied homozygous tall and heterozygous tall. This homozygous and heterozygous refers to which one? Genotype. So during mutation, there is a change in the DNA sequence. So automatically first the genotype changes, that leads to change in the phenotype of an organism. This is called mutation, and this mutation always leads to genetic variation, differences in the genetic makeup. It always leads to genetic variation. So mutation is a sudden inheritable type of change. This change is due to changes in the base sequence. Whenever change occur, first the genotype will change, then automatically phenotype changes, and mutation leads to variation. Mutation always will leads to variation. Now let us pass on to what are the types of mutation. There are two types of mutation. One is called point mutation and the second one is called as frame shift mutation. One is of point mutation, second type is of frame shift mutation. If the mutation is because of change in a single base pair. Got the meaning of base pair? Last year when you have studied about the structure of DNA, you have studied adenine combines with thymine by 2 hydrogen bond. This is a single base pair. Guanine combines with the cytosine by 3 hydrogen bond. That is a base pair. So, if the mutation is due to change in only one base pair, then it is called a point mutation. Example for point mutation is of sickle cell anemia. About sickle cell anemia, in the later part of the lesson, you will read in detail. Second is called a frame shift type of mutation. Here, either deletion or addition of base pair occur in a DNA. That is because of change in only one base pair. Here it is change in more than one base pair. That is either addition of more than one base pair or deletion of base pairs of DNA. That is called a frame shift type of mutation. Example is of Tay-Sachs disease. So first you have studied what is mutation. Second is types of mutation. First type is point mutation due to change in single base pair. Example, sickle cell anemia. Second type is frame shift mutation. This is because of change in more than one base pair. That is addition or deletion of base pairs of DNA. Example is of Tay-Sachs disease. Now, next one is about mutagens. What are mutagens? These are the agents that causes mutation. The agent that is inducing mutation is called a mutagen. These mutagens are being classified under three categories. First category is a physical mutagen. Example is of X-ray, UV rays and gamma rays. These rays, when a person is exposed very frequently to these rays, they can cause mutation. Second is of chemical mutagen. Example is of nicotine. Example for chemical mutagen is of nicotine. Third is a biological mutagen. Example is of virus. Sometimes virus also can cause mutagen. Virus is a living entity. It's a borderline. So, it is considered to be a biological mutagen. So, mutagens are the agents that are causing mutation. One is physical. Example, X-ray, UV rays and gamma rays. Chemical mutagen, nicotine. And third is of biological mutagen is of virus. Now let us pass on to genetic disorders. So the term genetic disorder. So you have studied in your lower classes about contagious disease, non-contagious diseases. Now you will be studying about genetic disorder. So these disorders are inheritable, that are passed on from parent to offsprings. These disorders are broadly classified under two categories. First category is of Mendelian disorder and the second category is of chromosomal disorders. Clear? Genetic disorders are broadly classified under two categories. One is Mendelian disorder, other one is of chromosomal disorder. First, let us study about Mendelian disorder. Here, a change in a single gene. The disorder is because of change in a single gene. It always follows Mendelian pattern of inheritance. By now, you know what is Mendelian pattern of inheritance. Mendelian pattern means the dominant allele will suppress the recessive one and express itself in the first filial generation. And these disorders can be easily identified by pedigree analysis. So what is a pedigree analysis? It is a tool to find out how far the characters are moving from parent to offspring in a human family. In detail, you will be studying at the later part of the lesson. So, pedigree analysis is a tool meant for the analysis of characters, how far a particular trait is inherited in a family. Clear? So, Mendelian disorders are because of change in only one gene. It always follow Mendelian pattern of inheritance. It can be identified by means of pedigree analysis. Example for Mendelian disorders are of one is hemophilia, cystic fibrosis, sickle cell anemia and color blindness. In detail, you will be studying about all these diseases. Hemophilia is called royal disease. Second is of cystic fibrosis, sickle cell anemia and color blindness. These are examples for Mendelian type of disorder. Second type of genetic disorder is called a chromosomal disorder. So here the disorder is because of absence of one or more chromosome or due to the presence of excess of one or more chromosome. Every species is having a particular number of chromosome. If you take human population, we have 23 pairs of chromosome, right? So either absence of one or more chromosome or excess presence of one or more chromosome lead to which disorder? Chromosomal disorder. So here aneuploidy. What is aneuploidy? Aneuploidy means failure of separation of chromatids during cell division either result in gain or loss of chromosome. The chromosomal disorder is because of which phenomena? Aneuploidy. What is aneuploidy? During cell division you have studied the spindle fiber will pull the chromatid. That time the half of the chromatid will move towards one polar region, other half will move towards another polar region. During which phase? Anaphase stage. But aneuploidy is due to failure of separation of chromatid during cell division that may result either in the gain or in the loss of chromosome. So loss of chromosome means if it is 2n minus 1. 2n here refers to in human population 2n is of 46 chromosome. So in monosomy the person will be having how many chromosome? The person will have 45 chromosome. Or sometime triasomy. Triasomy means 2n is 46 plus 1 that is of 47 chromosome. So chromosomal disorders are either due to the absence of one or more chromosome or due to the excess presence of one or more chromosome. So which causes this chromosomal disorder? Aneuploidy. Aneuploidy is failure of separation of chromatid during cell division. So it may cause either monosomy. Monosomy means 2n minus 1. Our 2n number is 46. So 46 minus 1 is 45. Triasomy means one extra chromosome, that is 2n plus 1, that is of 47 chromosome. This disorder is called chromosomal disorders. So let us study first Mendelian disorder. One example for Mendelian disorder is of color blindness. Color blindness is an example for Mendelian disorder and it is a sex-linked recessive disorder. Why we are using the word here sex-linked? Sex-linked means what? It is a X-linked disorder. X chromosome is having the gene for this disorder. The defect is due to mutation in certain genes present in which chromosome? X chromosome. That is why it is called a X-linked or sex-linked disorder. So this disorder is caused by which one? Recessive allele. Under which condition recessive allele will express? Only under homozygous condition. This is clear. Color blindness. It is X-linked or sex-linked disorder. The disorder is because of a recessive condition. Here the gene for the disease is located on which chromosome? X chromosome. The affected person cannot differentiate which color? Red and green color. That is why it is also called as red-green color blindness. Clear? They cannot differentiate red and green coloration. And it occurs in about 8% of male and 0.4% of female. So it is common among whom? Among the male. Always remember, sex-linked recessive disorders are more common in male than in female. Why it is common in case of a male? Because in case of male, why it is common? Male is having one X and one Y chromosome. Clear? They are having only one X chromosome. So even if this one carries the allele for color blindness, the male will be affected. But female is having how many X chromosome? Two X chromosomes are there. Even if one X chromosome carry the allele for color blindness, she will be a carrier. Externally, she will not show any symptom. That is why color blindness is seen in case of 8% male and only about 0.4% of female. Got it? Because it is a X-linked disorder, recessive disorder, X-linked. Because male is having only one X chromosome. If that carries the allele for color blindness, male will be affected. But female is having how many X chromosome? Two X chromosomes are there. Even if one is having the allele for color blindness, then also she will be a carrier. That is why it is common in case of male, less common in case of female. Go through up to this. Thank you all for your patient hearing.